Today, we're diving into one of the biggest, most expensive problems in all of medicine. How do you get a new drug from just an idea in a lab into the hands of a patient who needs it? You'll see it's always been a high-stakes gamble, a gamble that AI is now completely changing the rules for.
So let's just start with the stakes, shall we? This number right here, $2.6 billion. That's the estimated cost to bring a single new drug to market. It's an absolutely mind-boggling figure, and it's driven by one simple, really brutal fact.
And that fact is failure. Just think about this for a second. For every 20 drugs that make it into human clinical trials, 19 of them will fail. That is a staggering 95% failure rate. All that time, all that money, and all that hope, just gone.
So that brings us right to the core question we're going to answer today. Why is this so incredibly hard? The answer isn't just about finding a compound that works, it's about finding one that can survive the unbelievable complexity of the human body.
I want you to think of the human body as the ultimate obstacle course for any potential drug. And this course, it has a name, ADMET. It's this brutal biological gauntlet that every single compound has to run. And frankly, it's where most of them get knocked right out of the race.
So ADMET, it stands for 5 critical hurdles. That's absorption, distribution, metabolism, excretion, and toxicity. Here's the kicker. If a drug fails on just one of these, it doesn't matter how perfectly it hits its target, it is game over.
Okay, so let's just quickly walk through this journey. It all starts with absorption. Can the drug even get into your bloodstream to begin with? Then there's distribution. Does it actually travel to the right place, like the brain, or does it get stuck somewhere useless? After that comes metabolism and excretion. How does the body process the drug, and maybe most importantly, how does it get rid of it safely?
Now that metabolism step is super interesting. This is where your liver basically acts as the body's main chemical processing plant. And the key workers here are this big family of enzymes called cytochrome P450, or CYPs for short. These little guys metabolize something like 90% of all drugs on the market.
And this is exactly why metabolism is so tricky. You've probably heard that you shouldn't drink grapefruit juice with certain medications, right? Well, this is why. Grapefruit juice actually blocks one of those key enzymes, CYP3A4. And when that happens, other drugs you're taking can build up to dangerous levels. So you can see why predicting these interactions is absolutely essential.
And that brings us to the final, and by far the most dangerous hurdle, toxicity. This is the nightmare scenario, where a drug passes all the other tests, but it turns out to be poisonous, causing things like severe liver damage or heart problems. In fact, it's the #1 reason drugs get pulled from the market, even after they've been approved. So how do scientists actually measure this margin for error? Well, they use something called the therapeutic index. It's basically just the gap between a dose that helps you and a dose that hurts you. A really wide index is great. That means the drug is pretty safe. But a narrow index? That means the line between helpful and harmful is razor thin, like a tightrope walk.
You know, for decades, the only way to find these ADMET problems was through really expensive and slow animal and human trials, often way too late in the game. But that has all changed. We are officially in the era of prediction.
Our ability to predict these problems with computers has really evolved through 3 distinct phases. First, we had these pretty simple rule-based systems. Then came what we call the data era, where just having the most data was king. And now, we're right in the middle of a full-blown deep learning revolution.
So in that first era, one of the biggest tools was something called Lipinski's Rule of 5. Now, this wasn't some fancy AI, it was just a simple set of guidelines based on looking at what successful drugs had in common. It acted like a first-pass filter, quickly weeding out molecules that were probably too big or too greasy to ever be absorbed properly as a pill. And the impact of this new fail early, fail cheap philosophy was, well, it was profound. This chart says it all. Back in the late 90s, almost 40% of clinical failures were because of bad ADMET properties. But by the early 2000s, after these simple screening rules were widely adopted, that number just plummeted to 11%. A huge deal.
All right, so the next phase, the data era, was really powered by this explosion in our ability to generate and store massive amounts of experimental data. Algorithms like random forest became the standard, but they weren't the real secret sauce.
The crucial point from this era is right here. What mattered most was the data itself. A company with millions of high-quality, in-house measurements had a massive advantage. Their private data was worth way more than any special algorithm because, at the end of the day, better data simply builds better models.
And that brings us to today, the deep learning revolution. And this is a huge shift. This isn't just about getting better at predicting which drugs will fail anymore. This is about actively designing molecules that are destined for success right from the very beginning.
Okay, the key technical leap here is in how computers see molecules. The old way used these fixed fingerprints to describe a molecule's features, but the new way, using things like graph neural networks or GNNs, treats a molecule like the network it actually is. The GNN then learns for itself which parts of the molecule are most important for predicting something like toxicity. It's so much smarter.
Another huge breakthrough is something called multitask learning. So instead of training one AI model to predict, say, absorption and a different one for toxicity, you train one single powerful model to predict everything all at once. This lets the model share what it learns across different tasks, and that can boost accuracy by up to 15%, especially for properties where we just don't have a lot of data.
So, what does this all add up to? It's a completely radical change in strategy. The old way was just brute force, make thousands of molecules and just hope a few of them survive that ADMET obstacle course. The new way is generative design. You literally tell the AI the exact ADMET profile you want, and it designs a brand new molecule from scratch, one that's built to succeed.
And look, this is not science fiction. This is happening right now. Companies like In Silico Medicine are already using this approach to take AI-designed drugs from a simple concept all the way to human trials in under 3 years. That's a process that used to take nearly a decade.
So what all this really means is that drug discovery is transforming before our very eyes. We are finally moving away from a multi-billion-dollar game of chance, one that's just riddled with failure, and we're moving towards a predictive, design-focused game of science, where the odds are finally shifting in our favor.
And that leaves us with a really fascinating question for the future. We've just seen how AI is starting to tame that ADMET beast, making drug development safer and cheaper. So, if we can now design the perfect molecule, what is the next great mountain for medicine to climb?